joined us today. I look forward for the productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for the continuous professional development in pharmaceutical sector. I am confident that today's session will uphold our tradition of excellence, providing valuable insights from our respected speaker. So without wasting time, let's seize this chance to gain new knowledge and participate actively in progress in our respective field. We are gathered here to commemorate on 97th SPSR webinar on expediting drug discovery and development using AI, opportunities and challenges. A distinguished event hosted by the Society of Pharmaceutical Sciences and Research under the esteemed guidance of Dr. Shashi Alok sir, National President SPSR, continues to play a pivotal role in fostering innovation and advancement in the field of pharmaceutical science. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. Well, we make it a priority that we recognize and honor pharmacists across notable contributions who drive in our field. So at the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. Well, it's a great privilege to introduce Dr. Rajnish Kumar sir, Associate Professor, Department of Pharmaceutical Engineering and Technology, Indian Institute of Technology, BHU, Varanasi, India, as a distinguished speaker. Dr. Rajnish Kumar sir's expertise greatly contributes to our event, providing substantial knowledge and insights. His participation is certain to enrich our discussions and expand our knowledge and horizons on expediting drug discovery and development using AI. We are grateful for his engagement and anticipating an enlightening session ahead. So it's also very important to introduce, privilege also to introduce Mrs. Monika Sabharwal, ma'am, as a convener for today's webinar, who also serves as the founder and national secretary of SPSR and CEO, SPSR Periodicals Private Limited. Significant involvement and leadership in the pharmaceutical sciences research community are indicative to our commitment in the field. We are very grateful to have a guidance in today's session also. Uh, Without further ado, I would like to go through guest profile, short profile of our speaker. So, Dr. Rajnish Kumar sir is a distinguished academic and researcher currently serving as an associate professor in the Department of Pharmaceutical Engineering and Technology at Indian Institute of Technology, BHU, Varanasi. He earned his PhD in pharmaceutical sciences from the University Institute of Pharmaceutical Sciences, Punjab University, Chandigarh, where his doctoral research focused on synthesis and evaluation of steroids as 5-alpha reductase inhibitors. Following the completion of his PhD in 2014, Dr. Kumar joined the prestigious Karolinska Institute in Stockholm, Sweden, where he worked for over 5 years as a postdoctoral research associate at the Center for Alzheimer Research under the mentorship of Dr. Tahir Darasat and Professor Bangit. His research there was centered around neurodegenerative diseases, particularly Alzheimer's disease. Dr. Kumar has also been awarded the highly competitive Japan Society for the Promotion of Science Summer Fellowship, during which he collaborated with Professor Kenzie at the National Institute of Biomedical Innovation, Health and Nutrition, Osaka, Japan. His current research interests include the integration of machine learning tools for ADME prediction and the design and development of small molecule therapeutics for various neurological diseases. With a strong background in both experimental and computational drug discovery, Dr. Kumar is contributing significantly to the advancement of pharmaceutical sciences and translational medicine. So it's with a great satisfaction that we have declared that today we have exceeded 1,500 registrations for this webinar. Registrations received from India, Albania, Algeria, Bahrain, Egypt, Ethiopia, Indonesia, Kenya, Kuwait, Malaysia, Nepal, Nigeria, and the list goes on, Vietnam and Zimbabwe also. We are deeply moved by the substantial and the very responses we have garnered. Our heartfelt gratitude goes to each one who has, each person who has taken a moment to participate in this significant event. So once again, thank you for all joining us today. So let's make this webinar impactful. We have our esteemed keynote speaker, Dr. Rajnish Kumar, sir, with us. Welcome, sir. Your insights will be highly valuable. Uh, Over to you, sir.
Yeah, thank you so much, Dr. Junaid. Thank you so much for inviting. I'm grateful to the SPSR for this chance to interact with, researchers across the world, and especially on this topic. So I would like to share the screen with all of you, and then we will start. Okay, so today's session is on the role of artificial intelligence, how we can expedite the drug discovery and development using artificial intelligence. So I probably know about this Nobel Prize actually. So this is the building which you see here in front of this, fountain is the Nobel Forum, which is where the Nobel Prize in Physiology and Medicine is being decided to whom the Nobel Prize is given. Because when Alfred Nobel in 1900, around 1900, when he wrote his will, his testament, so he gave this responsibility to Karolinska Institute that the Nobel Prize in Physiology and Medicine should be decided by Karolinska Institute. And I was fortunate to work there for almost 6 years. It was a great experience actually. So a little bit background of me. I did my PhD, as Dr. Junaid said, from Punjab University, Chandigarh, and then I was a postdoc in Karolinska Institute for quite a time. Like I was working in two different groups, where I was working with Professor Dareshwari on discovery of novel choline acetyl transferase ligands as potential PET tracers for the early detection of Alzheimer's disease. And then I was working with Professor Winbla in a project where we were trying to develop novel therapeutics targeting the protein-protein interaction, which is leading to decreased amount of tau aggregation and tau hyperphosphorylation. So then I was also fortunate to work at KI as an assistant professor, and then I was visiting the lab of Professor Kenji Mizuguchi at Osaka University, Japan, and at Nihon, Japan. And then since 2019, I'm working here at IIT BSU in the capacity of assistant professor. And then recently I got promoted to associate professor. And I'm associated with Department of Pharmaceutical Engineering and Technology, where, the main idea is to get the work on the discovery projects actually. And the Center for Alzheimer Research was working on different aspects of drug discovery. So there were people working on structural biology, and then neuroimaging, and then epidemiological studies. And it has three different campuses where the Center of Alzheimer Research is a hub for all the Alzheimer research within KI. And the center also provides a forum for innovative translational research with the aim to better understand, diagnose, treat, and cure the Alzheimer's disease. So coming to the topic, what is a drug? So I believe most of us are pharmaceutical or coming from pharmaceutical or aligned discipline. So a drug is a substance that is used to prevent or cure a disease or ailment or to alleviate its symptoms. While if we talk about this one sentence definition of drug, but there are like at least 4 different definitions are hidden in this one sentence actually. So a substance can use to prevent or cure a disease. So the substances which are being used for preventing the disease, those are called as, prophylactic treatment, like the folic acid tablets, or the vitamins which people are taking so that they do not get ill, like folic acid is taken, or the iron supplements are taken so as to not get the anemia, right, and not get anemic. Or to cure a disease means like where we have, for example, the substance which we are using to cure a disease, those are called as, you know therapeutic actually. So they are therapeutic in nature, they are providing the treatments, okay, curing a disease. And then you have another definition which is like alleviate its symptoms, where we are using not only to treat the disease, treat the cause of the disease, but to alleviate the symptoms, like we are using uh, you know the paracetamol in case of we are getting the fever. So once we get the fever, we use paracetamol, and that paracetamol is not curing the disease, but it is just, alleviating the symptoms, like it is reducing the fever. We get rid of the fever, but we do not get rid of the disease. In case like we get a viral infection or bacterial infection, or due to some any other disease, we are getting the fever. So the drug discovery itself is a very challenging process, and we can divide it into discovery and development. So if we talk about the discovery, so then it's highly challenging, where you have to start with, identifying a target. So you select a target, and for that target, that target selection should it depends. We have to think about a lot of things, like whether the target is validated, whether the assay which we are using for validating or testing the target is, we have a robust assay system, or whether we have the structural information of the target available with us, like using the 3D structure determination methods like X-ray crystallography or single particle cryo-EM, or whether there are compounds which can inhibit this target. So this target selection itself is a very challenging thing. And once we identify a suitable target, then the next step is to identify inhibitors of that target. And then for identifying inhibitors, we usually use like this high throughput screening methods, where we screen large number of compound libraries. Like we take maybe like 50,000 compounds and screen them against a target. Or we use, like a computational screening method, where we use virtual screening, where we can screen millions of compounds using these tools, algorithms, and then we try to identify the inhibitors. And once we have identified in an inhibitor, it is called as a hit compound. Okay. Or we can also identify inhibitors using the natural products or the traditional medicines. Like this anti-malarial drug was identified from Chinese medicine by Tu Yu, who got the Nobel Prize in medicine as well. So once we have identified an inhibitor, so that inhibitor is, usually it is called as a hit compound, because it is binding to the target, but it is usually not very, strong inhibitor. So once we have identified an inhibitor, so we have to optimize it and test it, validate it. So once we validate it, these are called as validated hits, and then we get a lead molecule which has shown activity in the animal models and preferable optimal pharmacokinetic properties as well. And once we get a lead molecule, we have to apply the iterative medicinal chemistry approach to optimize its efficacy and pharmaceutical qualities. And this lead is then further developed into a drug candidate, where we get more information about the potency, efficacy, pharmacokinetic, and also early toxicity information as well. So you can see that it is highly challenging and a long process, where the challenge is that the success rate is very low. For example, only 2 to 5%. And it is quite costly, where you can see that it costs around 500 million US dollar. And then it takes a lot of time, like 3 to 5 years, just for the discovery phase. And then comes the development phase, where the drug candidate is now, evaluated in preclinical safety studies, and then it is detailed toxicity studies are are done, where the compound is tested for mutagenicity, detailed pharmacology, acute toxicity, pharmacokinetic or ADMET properties, subacute repeat dose toxicity, and also the according to the GLP studies, its manufacturing shall also be optimized and validated, where the formulations are developed and F&D is done, and then you get a good formulation which has a proper optimal pharmacokinetic properties and all. So once you obtain that, once you do that, then it moves to the clinical phase, where these are being evaluated in clinical phases like phase 1, phase 2, and phase 3. And then the objective of phase clinical trials is to evaluate its safety and efficacy actually. So in the phase 1, you study whether the compounds are safe, and then in phase 2, you start with evaluating the efficacy, and in phase 3, you do it in a larger group actually. And finally, once the compound clears all these phases, then it goes for the registration to the regulatory body like FDA in US. In EMA, there is European Medical Agency. In Japan, there is another agency. In India, there is CDSCO. So where once you get all the documentation done, so you apply for final approval from the regulatory agency, and once it is done, then the drug reaches to the market and then used for treatment of the patients. But not only that, even after approval, so some drugs, they might show toxicity, and those are called as adverse drug effects. And there are studies for evaluating those, and those are called as post-market surveillance, or they are also known as phase 4 clinical trials. So, and the development phase also is, the challenge is that success rate is quite low, and then it's quite costly, and it takes a lot of time. Okay, so just to emphasize on how difficult it is to discover a drug and how challenging it is, I just would like to share this case study of discovery of Omeprazole, which is a blockbuster drug having a sale of billions of US dollar per year. And then Omeprazole is probably one of the drug which almost every one of us has used at least once in our lifetime, because it's very like OTC drug, over-the-counter drug, which is used for treating the hyperacidity, reducing the acid secretion. So the company AstraZeneca wanted to discover a molecule which can be used for, developing this anti-secretory compound, or reducing the hyperacidity. So they started with this compound called CMN131. So they identified this compound, which was having a potent activity in inhibiting the acid secretion. And then what they realized that when they tested this compound, so they found that this compound is, showing liver toxicity. So then it was not possible for this compound to move forward. So then they tried to convert this molecule into another molecule, and then they developed this molecule called H124/26 in 1973. Okay, so then in this molecule also they had this problem where they found that this molecule was already patented by another company, so they could not use this molecule for their research. They had to further optimize this molecule into another molecule, and then they identified timoprazole, which was H83/69, which was a metabolite of H124/26. However, this molecule was also showing adverse effects where it was, inhibiting the iodine uptake. Okay, so this molecule was also not able to taken further. So then they had to identify another molecule. So they got this molecule called picoprazole, H149/94. So with this molecule also, they had this necrotizing vasculitis, which was a severe side effect leading to the severe toxicity in case of this molecule. So they could not further take up this as well. So after all these failures, after all these challenges, finally in 1979, so the company was able to find the molecule Omeprazole, which was a potent and non-toxic molecule. So you can see, like, starting from one molecule and having several failures, finally they were able to solve this riddle. However, this was done in an industrial setup where there were like hundreds of researchers working on a single project with, means ultimate amount of funding without any issues. So then they were able to identify, develop this molecule. So you can see like how challenging drug discovery is, and how difficult it is actually. So not only that, because the patent life is limited. So once they identified this molecule omeprazole, so they had to, develop another molecule to stay in the market. So they developed S-omeprazole, which is S-enantiomer of omeprazole, which was found to be more active. So then they marketed this molecule. So with this case study, you can see like how challenging or how difficult, drug discovery is. And the idea is like how we can speed it up by using artificial intelligence. So now we will quickly look at what is the artificial intelligence and how we can use it for making drug discovery faster. So in this case, you can see like there is a number called 2, and then we have the 4. So then we have 6, and then we have 8. So now anyone can tell what will be the next number, right? So after looking at this series of number, like every time we are adding 2 here, so we can like with very high confidence we can predict that next number will be 10. Okay, so that is a quite easy job that we can do. But what about this problem? So which of the following molecules are soluble in water? So by just looking at the structure of this molecule, until and unless there is someone who is a medicinal chemist and working in industry for like several decades. So by looking at the structure, they can probably tell, but still it is a very difficult problem that we cannot solve, right? So what was the reason why we are not able to solve this problem while we were able to clearly tell the answer to the earlier problem? Because in that series of numbers, we could see the pattern, we could identify the pattern very easily. But in this case, we cannot identify the pattern, right? And this is just a small, like very few molecule. If I give you structure of maybe like 10,000 molecules and ask you which of the molecule can be soluble in water, so then that will be even a difficult problem where we will not be able to see the pattern, understand the pattern, right? So the idea of using computers or AI or machine learning in these kind of problems is because the computers are faster, right? They have a lot of speed. They can do the calculations faster, and then they work 24 into 7. So it's not like us that we have to take rest during the night and then we can work. So this is how the machine learning or AI can help us in identifying the patterns, identifying the problems, and then solving them. So looking at the overall structure of AI, so everything which is nowadays used is comes under the umbrella of AI, and because these are some knowledge-based systems where we use them for solving the problems. And then if you go a little bit deeper, where logistic regression is used, for example, those methods are called as machine learning methods. And then if we are using shallow autoencoders, which are further a subset of machine learning, these are called as representational learning. And then if we go further deeper, like we are using multi-level perceptrons, MLPs. So this technique is called as deep learning. Especially nowadays, deep learning is quite popular, especially due to development of all those tools like whatever we are using nowadays. Okay, so what machine learning is? So the machine learning is basically what we are doing here is we are using known data and develop a model to predict the unknown data. So where the known data could be the big enough archive or the previous observations or the past data, and the model is like the known data and the algorithms, which are basically mathematical algorithms. And then the unknown data could be the missing data, unseen data, or the non-existing or the future data. So it is like predicting the temperature or the chances of rain over next 1 week using the data from previous 1 month. So for previous 1 month, the meteorological department, it collects the data related to relative humidity, wind speed, wind direction, temperature, and then the movement of clouds using satellite images, and putting all that data into predictive model algorithms, we can predict what will be the chances of rain in future, in upcoming days, right? So this is like we can use it in drug discovery and development. Like we can use the historical data, like for example, solubility of compounds. If we have a set of 1000 molecules whose solubility is known to us, so then we can use those properties, that data, and then we can make a model which we can use to predict the solubility of unknown molecules. So coming to the topic, like how AI can help to accelerate drug discovery and development. So there are multiple ways, and starting from the hit identification to the clinical trials. So AI can help us in all the stages. So let us see one by one. So the first thing we can do with AI is we can identify the targets and validate them. So by identifying by knowing more about the protein ligand interaction, and then by prioritizing the targets using the AI algorithms. And then we can identify hit molecules by virtual screening of the compounds, or we can also generate molecules using the generative AI, where we can design the compounds from scratch. That is also called as de novo drug design. And then we can use it for predictive toxicology and safety assessment, where we can use it for in silico ADMET prediction, and we can do the toxicity modeling with the help of AI. And then we can, do the biomarker identification and patient stratification. That is how we can use AI in the clinical trials. So we can use AI in genomics and proteomics, dealing with the data coming from, the big data coming from genomics and proteomics studies. And we can also use it for personalized medicine advancements. So let us see how people have been using AI for target identification. So this is a study from in silico medicine, the company called in silico medicine, which is one of the leader in using AI for drug discovery. So in this study, what they did was they have developed a tool called Pandaomics. So by using that Pandaomics, so they could deal with all the multi-omics data, for example, genomics, transcriptomics, proteomics, epigenomics, metabolomics, signaling pathway, molecular interaction, protein-protein interactions, and the text data coming from grants, patents, publications, clinical trials, and financial reports. And by putting all this data into this AI model, which is literally a LLM-based model, so it could identify some potential targets, and then those potential targets can be selected on the basis of these criteria, like disease causality, developmental status, like clinical trial phase or ligand compound availability, or druggability, like whether the compound is druggable, protein localization, therapeutic modality, availability, protein structure, toxicity, gene essentiality, tissue specificity, cellular process involved, and novelty. And then you can come up with a target of your choice. So this is how, because uh, otherwise it is very quite difficult to analyze that big data coming from all these, diverse data coming from multiple, technologies. But with the help of these LLMs and with the help of AI, we can do that. So this is how they discovered a molecule called INS 018-055 for the treatment of idiopathic pulmonary fibrosis. So they analyze all this multiomics data. They use this iPanda from their tool actually, and then with the help of this tool, they came up with 20 different targets for validation. So once they identified these 20 targets, so they prioritize top novel intracellular targets out of these 20 targets. And then by using generative AI, de novo compound generation, so they generated several new molecules, which were quite potent, like nanomolar with nanomolar potency. And then they evaluated those into these primary assays, and then they did specificity and selectivity assays. And then they did, collagen production assays and fibrosis assays. They performed in vivo studies, fibrosis in vivo studies. And finally, they identified those molecules, which are now one of those molecules called as Rentosertib is now in phase 2 clinical trial. So that is how they could achieve, they could discover a small molecule starting from scratch, discovering a new target, discovering new molecules, and that too in very limited time. So it took them only 18 months actually to take that molecule to the clinical trials, starting from scratch, identifying a new drug target as well. So this is how AI can speed up the process of drug discovery. Okay, so the second step is like you can use it for hit identification. Okay, so this is one of the study which we also performed using the tool called Deep Doc. Okay, so in this tool, what can be done is like, for example, if you have an ultra large library, like billions of molecules, this library is around 1.3 billion molecules. If we talk about Zing 20 library, so you have 1.3 billion molecules, and then those 1.3 billion molecules, screening them using structure-based method is quite difficult. So you cannot use them. So by by taking a small chunk of this library and then converting them into fingerprints and docking them and getting their docking score. So what we did was we developed a model which can predict the docking score. So we use deep learning to develop this model which can predict their docking score, and then this model was further used to predict the docking score of this huge 1.3 billion molecules library, ultra large library, and then so that we can prioritize the compound. So this is how we can use AI for identifying hit molecules as well, which is the second step in drug discovery and development. Okay, another way to generate new molecules is the generative modeling. So this is a question I asked to ChatGPT. So can you suggest some journals in the field of AI and drug discovery? And it was very good at it. So it could suggest the journals which are publishing in this field. But when I asked this ChatGPT to generate molecules, like can you generate structures of new drug-like molecules that are permeable to blood-brain barrier? So it was not able to do that because this tool is trained to generate the text, not the molecules. Okay, but there are tools which have been developed by people, the researchers all globally. So these tools can generate novel molecules which can be used to generate drug-like molecules actually. So this de novo molecular design is basically what it does is it automatically proposes novel chemical structures meeting desired molecular profiles to elicit a desirable biological response while maintaining acceptable pharmacokinetic properties. These are also known as generative chemistry, which reflects the rise of generative models in AI. And these models are trained on large data sets and learn to produce output that is similar to the examples they were trained on. Okay, so this is, I would like to skip this. So I come to this actually. So there are basically two approaches to design a molecule. One is called as direct approach, where we try to, for example, synthesize a molecule and then we experimentally determine the properties, or we, do the simulation and determine predict the properties, and we see where this molecule lies. So this is a functional space. For example, functional space representing the desired properties, like in this case, redox potential, solubility, or toxicity could be there. And then this is the chemical space where we see, because this chemical space is huge actually. So there are billions and trillions of molecules are there which can be made, okay, and then which can be explored. So by using the direct approach, we can only we have to synthesize and develop molecules one by one, and then we have to either experimentally or computationally determine their properties and see until and unless we are there in our desired chemical space, functional space. So another approach is the inverse approach, where we have those molecules already with us, and then we have their properties, and then we filter them out according to the properties, and then we try to identify the molecules which are in desired chemical space. This is called as inverse approach, and this is like the virtual screening or the high throughput virtual screening which we uses. And then another inverse approach is the generative AI, where we start with one molecule anywhere in this chemicals in this functional space, and then we try to optimize that molecule, okay, step by step to reach to the desired functional space. So this is where the generative AI comes into picture. And then there are multiple models, like there are variational autoencoders, generative adversarial networks, or reinforcement learning, which have been used for generating these molecules. So Reinvent 4 is one of the tool which is developed by AstraZeneca. So in this tool there are multiple generators where Reinvent can generate atom by atom generation. It can generate molecules by atom by atom. And then there is libinvent, which can be used to generate the library, design the library and scaffold decoration. There is link invent, which can be used for fragment linking and scaffold hopping. And then there is mole to mole, which is a transformer based molecular optimization method. And then you can use multiple scoring functions, like you can use an RDKit function to predict the properties and use them as score. QSAR can be used, and then Pharmaco4 model can be used, and ChemProbe can be used, where you can predict the properties of molecule using the ChemProbe. And then it has a generator. Okay, so these are different generators. It generates a molecule, and then you can use it for transfer learning or reinforcement learning as well. Okay, and of course the scoring, as I showed here, that you can use the scoring. So the idea is that the generator will generate a molecule, and then it will be scored. For example, if you are using the reinforcement learning, so it will be scored, and then once it is scored, it will produce the scoring, and then this scoring will be fed into this model. Okay, so that it will, based on the scoring, it will generate similar molecules or dissimilar molecules, whether the scoring is better or not. So this is how it can generate novel molecules using the generative AI. So this is an example where the company called, again, the In Silico Medicine. So they used their generative AI tool called Chemistry 42. So Chemistry 42 is their generative AI tool. So which uses the model Gentrel, GENTRL model it uses. So by using this, so they could generate molecules which are highly potent, like 10 nanomolar efficiency and 20 nanomolar efficiency against these targets. So this was against DDR1, and then this was against the CDK20. So they were able to develop molecules, generate molecules against these targets using the Chemistry 42 platform, which is a generative AI based tool. Okay, coming to the next step, which is in the drug discovery development, which is the ADMET optimization or lead optimization, where toxicity is one of the major issue. So there are multiple tools which are being developed for predicting the toxicity of compounds. So SafeAt AI is a collaborative initiative led by CDER, where NCTR is developing a suite of deep learning based QSR models for various safety endpoints critical to regulatory science and the IND review. So it focuses on 5 key safety endpoints like hepatotoxicity, carcinogenicity, mutagenicity, nephrotoxicity, and cardiotoxicity. So SafeAt AI facilitates drug safety research with a novel deep learning architecture that improves the precision in toxicity assessment by tailoring prediction to chemical characteristics. And examples of AI tools for assessing drug discovery profiles include DeepDili, DeepCarc, and DeepMS. So the DeepDili is a deep learning framework for predicting the liver toxicity. So it is generated by conventional machine learning algorithm with deep learning framework based on mole2 descriptors. So you have the molecules, and then different models were developed, and then to make final model. And then this model can predict whether a molecule will be liver toxic to the liver or not, where DILI is the drug induced liver injury. Okay, and then this is how this model was built. So in this picture you can see all the detail of the model building process actually. And then DeepCarc is a deep learning framework for prediction of carcinogenicity. So the molecules, the carcinogenicity is one of the major toxicity which needs to be evaluated before using any molecule into any formulation or into any drug development process actually. So in this case, they took those molecules, like around 863 compounds, and then they developed these models using KNN, LR, SVM, random forest, and XGBoost algorithms, and then they could classify those molecules into carcinogenic or not carcinogenic. And then there is this tool called a skin doctor, so which is a ML-based model for classification of small organic compounds into skin sensitizers and non-sensitizers. And the core of skin doctor CP is a random forest binary classifier that is enveloped in an aggregated Mondorian conformer prediction framework, and it allows users to define error significance levels as well. And it is trained on a curated data set of 1285 compounds measured in the local lymph node assay. So this is how you input the molecule, and then this skin doctor can tell whether the molecule is a skin sensitizer, non-sensitizer, or there is no prediction possible. So this is how the aggregated Mondrian model is working. Okay, and then another tool is DeepHit, which is a deep learning framework for prediction of Hz-induced cardiotoxicity. So it consists of three optimized deep learning models like descriptor-based DNN, fingerprint-based DNN, and graph-based GCN model. So here you can see the schematic overview of DeepHit, where the molecules in the form of smiles, those are being used, and then the molecular descriptors are being calculated. And then descriptor-based deep neural network model is developed, which can predict whether a molecule is Hz positive, Hz inhibitor, Hz blocker, or non-Hz blocker. And then the same molecules are then utilized for calculating the fingerprints and then developing the model using fingerprints. And then those molecules are further converted into graphs using RDKit. And then GNN-based graph neural network-based model was also developed to predict whether a molecule is Hz blocker or non-Hz blocker. Okay, and then the predictive ADMET, and as I told, like I was fortunate to work with Professor Mizuguchi. So Professor Kenji Mizuguchi at National Institute of Biomedical Innovation, Health and Nutrition. So their team is, developing a platform called as DrewMap. So this was a picture from back in 2019. And then their team is developing this model called DrewMap, drug metabolism and pharmacokinetic. Okay, and this tool, they have incorporated several predictive models, like this one, like computational model to predict the fraction of unbound drug in the brain. Okay, so what they have been doing is, for example, in this case, they use this data set from Campbell and Keck drugs, and then they calculated several descriptors like CDK descriptor calculator, Moodrad, and then pedal descriptor, and then they developed several ML algorithms, use several ML algorithms like random forest gradient boost, SVM, and radial functional kernel, linear kernel, and partially square. And then they validated those models using squared correlation coefficient and the root mean square error. And then they came up with a good model which can predict this property fairly. And then they deployed this model in this tool called DrewMap. So now there is another version, I believe 1.2 is there. So one can just search DrewMap into Google, and then they can go to this web app, and then they can predict the properties. So if you click on new prediction, so you can draw the structure, and then you can predict the properties of these molecules. So coming to the last part, which is the role of AI in clinical trials. So as I said, like you can use AI in all the steps, and clinical trial is also not untouched by this, because when a drug is failing in the clinical trial step, which is the last step, so the company is losing a lot of money because they have put a lot of time, a lot of efforts, a lot of manpower, a lot of money on this project. And if a drug is failing in late stage clinical trial, then it is very risky for the company and very difficult for the company to continue actually. So that is why if we can predict how well a drug will perform in the clinical trial. For example, if we can classify the patient and the clinical trial participants into classes like responder and non-responder. So if we can tell which of the person will, which of the person will respond to the drug, or which of the person will not respond to the drug. So then you can have a chance of running a clinical trial with large success, or you can predict the success of this clinical trial. So there is this tool called PyTrial. So it's a Python package that implements various clinical trial tasks supported by AI algorithms. So it implements 6 essential drug development tasks like patient outcome prediction, trial site selection, trial outcome prediction, patient trial matching, trial similarity search, and synthetic data generation. So this is how it works. So it has, this is the PyTrial architecture. Okay, it has the data, patient data, and then it has the the data related to the drug, data related to the disease, and data related to the trial, and then it implement the ML model like GNN, transformer, or RNN, and then it has the evaluation pipeline where it based on the prediction, the ACC, AU, ROC, MSC, rank based on ranking, precision, recall, and DCG in generation, privacy, fidelity, and utility. So all these things are done, and then it can predict all these 5 properties, 6 properties like patient outcome prediction, patient trial matching, trial site selection, trial search, trial outcome prediction, and patient data simulation, and using 34 different ML algorithms, and it has 23 AML ready data sets as well. And not only that, it can be used, in all these phases as well, phase 1, 2 through phase 4 actually. So those were like quick recap of how AI can be used in drug discovery and development. So, and we know that everything comes with the limitations. So the let us talk about some of the limitations of using AI in drug discovery. So this is not a recent, but quite like in 2023 July, so almost 2 years ago, we saw a news where the Sumitomo schizophrenia drug discovered with AI tech fails in 2 phase 3 trials. So no matter how fast, how good AI is, there is no alternate to experimental evaluation in drug discovery and development, and anything can go wrong at any point. Okay. So in this case, for example, you can see that the drug failed in phase 3 clinical trial means the company has lost a lot of money and a lot of things in that project actually. So, and this is the Gartner hype cycle. So if we talk about the current stage of this AI technology, so you can see right now we are almost here at the peak of inflated expectations. Means we are expecting a lot from AI. Whenever we talk about AI, we say that it will do this, it will do that, and it will lose people will lose job, and everything will be like replaced by AI. So that is peak of inflated expectation. However, we will see a trough of disillusionment, and then hopefully there will be a slope of enlightenment, and then further there will be plateau of productivity. So finally, the plateau of productivity, once we see, then only we will see the real application of AI. And hopefully that time will come soon, maybe in a decade or so. We will see how we can use, or we can exploit AI for the benefit of mankind in the case of drug discovery and development. So talking about the limitations, so the first very big limitation is the data quality and availability, because we have quite limited data in drug discovery, and which is obtained from comprehensive and high quality data can be challenging, as experimental data is often limited or expensive to generate, and data heterogeneity is another problem, where the data is coming from diverse experiments, diverse labs, diverse people. So the diverse types of data, including genomic, chemical, clinical, need to be integrated. However, these data sets often come in different formats and have varying levels of quality. So it's quite difficult actually to club all the data and use that as it is. And then the biological system is quite complex, the complexity of biological system. So the biological system involves drug interaction are highly complex, and AI models must account for this intricate relationship within biological pathways, which can be challenging to model accurately. And then there is this multifactorial nature of this biological system, where the disease often have multifactorial causes, making it difficult to identify specific targets for drug intervention. And then the interpretability of model is also a problem, where it is considered as the black box nature. The many AI models, especially deep learning models, are considered black boxes, meaning their decisions making process are not easily interpretable. in drug discovery, interpretability is crucial for understanding the rationale behind prediction and gaining insights into the potential drug targets. And then the transferability of models, where the domain transfer is an issue. The AI models which are being evaluated or which are tested on, tested on one domain may not generalize well to another. Transferring knowledge between different therapeutic areas or disease can be challenging. And the data drift, where the change in experimental protocols or data sources over time can lead to data drift, affecting the performance of the AI models. And then, of course, the computational resources, like the computational intensity. We need a lot of computational power, especially for these deep learning models, because those are power hungry. So that is another issue. And then ethical and regulatory concerns are also difficult to handle, because we have to ensure that the AI is being used ethically, and also there are some bodies, regulatory bodies, which are a little bit reluctant or skeptical to approve the use of AI in all those fields that you might have seen in Europe or in other countries as well. And collaboration integration is also a difficulty which we are seeing, because the data which is lying with the large companies, especially in the case of drug discovery and development, the data is lying with the pharmaceutical companies, and those companies are not willing to share the data with academicians or other companies. And this non-collaborative approach is, what is a big challenge in the case of applying AI in drug discovery and development. And then cost and investment, because developing and maintaining AI-driven drug discovery approaches require significant financial investment. Smaller research institutions and startups may face challenges in securing the necessary resources, and especially the hardware, the infrastructure is quite demanding actually. So, coming to the conclusion, so AI is highly promising in accelerating drug discovery by contributing to every step of drug discovery process. And AI can improve the clinical success rate of drugs by designing better clinical trials, and AI can increase productivity and reduce cost by automating routine tasks. However, it faces challenges with data quality and model interpretability, making it a tough road to travel. So with those words, I would like to express my gratitude to Dr. Junaid and to Dr. Shashi for giving me this chance to interact with the participants of this session. And I hope that you have enjoyed the session. And if you have any questions, I'm quite happy to take those questions. And also, I would like to just share that I have created this course called as AI in Drug Discovery on NPTEL, and it has seen quite good response. And then I would like you also to join this course if you feel that this course can be used. Can be You can register for this course actually. So yeah, maybe I can just share. Yeah. So this is the course which I have created, and it is already tomorrow is the last day to register. So those who would like to learn more about how you can use AI for drug discovery and development, so they can sign up for this course. So in this course, we have I will be covering all the basics of drug discovery pipeline, the introduction to AI in drug discovery, fundamentals of AI and ML, AI in target identification, prediction, and validation, AI in high throughput virtual screening and lead identification, AI in lead optimization and drug target interaction, ADMET predictive modeling in drug discovery, AI in clinical phase, de novo drug design using generative AI, advanced concepts, case studies, and also hands-on sessions. So I have at least like 10 hands-on session in this course, where you will learn how to use those tools and implement those into your own projects actually. So with those words, I'm happy to take questions from you actually. Thank you.
Thank you so much, sir. Thank you so much once again, Dr. Rajnish Kumar, sir, for delivering such an impactful, insightful, and thought-provoking session on particularly on expediting drug discovery and development using AI, its opportunities and challenges also. You have provided us with presenting the complex ideas of this field with real world relevance are truly commendable, sir. We really deeply appreciate your knowledge, your time, and you have shared with us today. Of course, this will be the enriching one, and we will also highly recommend to the all our residents that they can also go for your course that so I'm NPTEL. So with this, sir, we have received some questions out of few we have received. So we'll go with the first question. What is AI currently transforming traditional drug discovery pipelines?
Yeah, so as we could see, that's a quite good, quite interesting question actually. So as we could see that the main issues with drug discovery and development is that it is very slow. Like it takes a drug to reach to the clinic almost like 10 to 12 years, depending upon the disease target as well. For some diseases, which for some diseases like Alzheimer disease, we do not have any treatment actually. So there is no disease modifying small molecule therapeutics for treating Alzheimer disease. However, the disease is like almost a century old. So for last 100 years, people are trying to develop treatment for diseases, but we are not able to do that, right? So the advantage of using AI is the first thing is that it can decrease the time taken by time taken for drug discovery. Okay, and another thing is because if it reduces the time from 10 to maybe 4 years, so then you are saving a lot of time, a lot of resources, a lot of money, as well as you are relieving the suffering of patients actually. So those patients who could not get their disease treated, if they can get the drug, so that is a wonderful thing which can happen. And as I gave the example of this in silico medicine, rentosertib, so they have been able to do it. So they could discover the drug in just 18 months actually. Not only the drug, but even the new drug target as well. So I think it is like by using ChatGPT, one can write an essay in very quick time. So likewise, hopefully we can discover the drugs very in very short time and with high success, so as to reduce the suffering of the patients actually.
Sir, one question, it is out of the topic because as we have a lot of registrants from all over country outside the globe.
Mm-hmm.
Because you have an interesting and inspirational journey, so this is from the personal side. Maybe someone of it may get some inspiration from this. When is the first time, sir, you decided that I'm very good in this, and how it started into in your mind that no, this is what I will be doing. So can you please brief us that particular moment when you got the inspiration that I will continue with this particular topic or this particular thing and to pursue it further, sir?
Yeah, thank you so much. Yeah, that could be, I think, quite like subjective actually. For example, when I was doing my master's at Guru Jambeshwar University, and one of the teacher was using, these basic multiple linear regression and to develop QSAR models, quantitative structure activity relationship models. And then I was like, I got fascinated by seeing how by using mathematical models, how we can predict these structures. It is like just showing your hand to someone and predicting the future of that person actually. So likewise, if we can predict whether this molecule is going to be soluble, this molecule is going to bind to this target, or is going to be permeable. So that fascinated me a lot. And then when I joined PhD, so I got the chance to work with Professor M.R. Yadav at MS University Baroda, and my supervisor, Professor Manoj Kumar. He was, very helpful and very good researcher. So he motivated me, and then we started with, all these projects, and we used different predictive models. And when I moved to Karolinska Institute, and there I saw that I can use like a supercomputer facility by using those clusters. And and another thing is that I saw a lot of trainings and workshops. I attended a lot of trainings and workshops and sessions, not only in person but in virtual mode and online. So I think you just have to identify what is your ikigai, for example, your what motivates you, and then just follow it. Actually, follow your dream and work hard on it, and then I think you can achieve it. Yeah. Thank you.
Thank you so much, sir. Now we'll come back to the questions from the topic. So one of the questions is, sir, how can, as you tell, how can AI aid in the early detection of drug toxicity?
Yeah, so that's again a very good question actually. So like we saw that if a molecule is, for example, there was a drug called rofecoxib. Okay, so rofecoxib is a drug which was withdrawn from market just because of its cardiotoxicity, because
Right.
it was approved and it was being used by patients for treatment of inflammation and pain, because it was a selective COX-2 inhibitor. But when several incidences happened, so then it was found that it is causing this toxicity. So the FDA it recalled it from the market, and then it cancelled its approval. So if we could have, by using those predictive models, if we could have predicted that toxicity earlier,
Mm,
even before the approval, so then people would not have suffered actually. Okay.
true.
And that is like thalidomide actually. So thalidomide, you probably know, everyone
Thalidomide was the biggest
thalidomide,
tragedy.
thalidomide story actually. So
Yes,
the
yes.
pregnant women which were taking that drug for treating the morning sickness, so that was causing the
Cocomilia,
yeah, so kids without limbs actually, born without
limbs,
limbs. So
absolutely.
those tragedies can be, reduced or can be avoided by using predictive modeling. And now all these regulatory bodies, they are making it mandatory. Like recently, FDA also released a notification that whatever is there, so first you have to prove by using in silico methods that these molecules are not toxic. So
Right.
first screen is always going to be in silico screen, and that is now all those FDA and all those regulatory bodies are coming up with actually. For example, these FDA is also reducing the animal uses in research. So can we develop some highly reliable predictive tools, predictive models which can predict the toxicity or other properties as well, so that we can reduce the number of animals being used in testing. Okay, so
Right.
I think if we can get a good data set, and then we can make good models, it's very highly likely that we can get a good tool which can be used for in silico prediction, early stage toxicity screening of those molecules, and that can help a lot with overall drug discovery and development.
Thank you so much once again, Dr. Rajnish sir, for answering all the questions so insightfully and patiently. Your responses have not only clarified the complexities of AI, but definitely also provide valuable perspectives for both budding researcher as well as professionals in the field. We truly appreciate your time and effort, sir. As we are going to conclude the session, the 97th SPSR webinar, we extend our heartfelt gratitude to our esteemed speaker, Dr. Rajnish Kumar sir, for his exceptional and insightful presentation on the role of artificial intelligence in accelerating drug discovery and development. Your deep expertise and practical perspectives, sir, has provided us valuable takeaways on how AI is revolutionizing pharmaceutical research from predictive modeling to lead identification and beyond. Yeah, so we thank all the participants for their active engagement and thought-provoking questions which enrich the session further. So we are deeply thankful to Dr. Shashi Alok sir, National President SPSR, for honoring us with his presence. I would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing the event. We extend our gratitude to all the SPSR members who are working behind the scene. So thank you. Your collective contribution have made this webinar a success, and we look forward for continuous collaboration in advancing the field. Stay connected with SPSR for upcoming webinars and initiatives. Until then, take care, stay safe, and continue with the journey of lifelong learning. Thank you. Have a wonderful day ahead. Thank you so much, sir, once again for being. Thank you so much, sir.
Thank you so much.